Okay, hello everybody. Welcome to what I think will be the last video in the basic cell biology and expanded targeted chemotherapy series. And this last video, we're going to cover what I think is the most exciting area of targeted therapy, and that's targeted cancer immunotherapy. The immunotherapy means that we're using the immune system, your body's immune system, and trying to get it to recognize and attack the cancer cells. Now, your immune system is made up of white blood cells, and it's the system of your body that's designed normally to protect your body against foreign cells and to some extent foreign chemicals, that is, cells and chemicals that are not supposed to be in your body. Your immune system is designed to recognize those cells and chemicals and actually attack them. If they're cells, they're attacked with chemicals designed to destroy cells that are produced by some of your white cells and then released. And if they're abnormal chemicals, they're white cells that just absorb them to get them out of your system and circulation. By the way, your immune system is also the system of your body that creates inflammation. Early on in an immune attack, your immune system produces chemicals that causes an area of your body to swell up with extra fluid and to get sort of red and warm. And what that is just a way of getting more white cells essentially to that area of your body so that they can battle whatever it is that's invading you. Um, your immune system is what protects you from germs like viruses and bacteria and from parasites. It's also what causes, rejection of organ transplants when your immune system recognizes the cells in that new organ as belonging to someone else, essentially foreign cells. It will attack them, and that causes a rejection reaction, which is unfortunate. And it's also the system that can cause you to die if you get the wrong blood type. For example, if you're infused with blood that's the wrong blood type, your immune system will recognize that type as not you because there are chemicals on the surface of these blood cells that are different than the chemicals on the surface of yours. And what your white cells do is they crawl around all over your body and they feel the surface of cells. They're concentrated in areas of your body where germs are likely to come in. So, for example, your skin, the tissue that lines the inside of your mouth and your stomach and your intestines and the inside of your genitals. These are areas with high concentrations of white cells because those are the areas where if you're going to get invaded by like a germ, that's where the germ is normally going to enter you. So the white cells there, they crawl around all over the place. They feel the surface of cells. And if they feel a cell surface that feels abnormal, that has chemicals in it that are not normal for your cells, for your body, then they send out all kinds of alarm chemicals which cause inflammation, but also stimulate white cells to start reproducing and to go on the attack. Now, the thing about cancer, which is interesting, is all cancer cells have abnormal surfaces. Some of the mutated proteins in cancer cells are cell surface proteins, often cell surface receptors, that are made abnormally or where there's too many or too few of certain types. And so normally your immune system would be able to feel those abnormalities, that there's way too much of a certain protein on these cells or that it's been made abnormally so it has the wrong shape and size. And so normally because your cancer cells have those abnormalities on the surface, your immune system, one of its jobs would be to destroy the cancer before it has a chance to kill you. And that's actually what happens. Most Cancer biologists believe that the average person probably gets cancer, 10 or more times in their life. But in most of those times, the immune system recognizes the cancer and destroys the cancer cells before there's a significant number of them and before they can, create large tumors and spread around the body to different organs. So the cancers that actually become a life or death problem for you are cancers that have cells that are not just abnormal in their growth abilities and their ability to move around the body, but they're abnormal in they've developed ways of protecting themselves against your immune system. So one of the hot areas of cancer research over the last 20 years has been figuring out how cancer cells protect themselves from immune attack, because the ones that haven't got that protection, they get destroyed by your immune system and they never become a serious threat to your health. So the cancers that become a serious threat to your health are the ones that have developed cells that can evade the immune system. So how do they do it? Well, we've done a lot of heavy research on this over the last 20, 25 years, and it turns out there's a number of different ways that cancer cells can protect themselves from your immune system. One way is by creating what's called the PD-L1 chemical. Now, most cells in your body, once you're out of your mother's womb, they take away their PD-L1 chemicals. And for good reason. That's because the PD-L1 chemicals are chemicals that can shut down your immune response. They actually shut down the response of any white cell that touches the cell with the PD-L1 molecule. So cancer cells that become a problem for you are the ones that have reactivated their PD-L1 gene. And so now they make the PD-L1 molecule, which means that they can deactivate pretty much any T cell that touches them by attaching their PD-L1 molecule to a receptor on the white cell called PD-1. And when those two things bond together, it just completely inactivates the cell. So remember I said that the way your immune cells detect cancer is by rolling over your normal cells in your tissues and feeling the surfaces for some abnormality. But when they touch the PD-L1 chemical and it attaches to their PD-1 receptors, which all white cells have, or most white cells have PD-1 receptors, then the white cells just get completely shut down. So as soon as they touch that cancer cell, they get shut down, which means they can't attack it or send an alarm to other white cells. And it turns out a significant number of really dangerous cancer cells have reactivated their PD-L1 gene. So one of the mutations they have is a mutation that allows them to transcribe and translate the PD-L1 protein, which, as I said, is normally shut off. This gene is normally shut off by most cells after birth. Okay, another way that cancer cells protect themselves, this isn't as common, is they can do what's called epigenetic cell type switching. And if you'll remember from when we talked about epigenetics, that epigenetics is a way for your cells to just, basically shut down the transcription of a number of genes so they can, become the kind of cell that they're meant to become, like a liver cell or a kidney cell or whatever. Different types of cells shut down different genes in the genome. And turns out cancer cells often develop the ability to change their epigenetic modifications to things like histones, and that allows them to change the type of cell that they are and completely change like their surfaces. And so that can fool your immune system into, thinking that this is a normal cell because it's now changed its surface into a different type of cell, which is, one of your regular cells. Like a cancerous liver cell may change its surface so that the surface chemicals are the chemicals of a kidney cell, but they're normal chemicals of a kidney cell. So to your immune system, it feels like a normal kidney cell, even though what it is an abnormal liver cell that's just reactivated a bunch of genes. That's not a very common way that cancer cells evade the immune system, but some do that. The PD-L1 gene activation is a very common way that cancer cells evade the immune system. Another way that's reasonably common is cancer cells often have mutated mucin proteins. Mucin proteins are the proteins that are in mucus. And, mucus is produced by the cells that line like your respiratory tract and your nasal tract and sinuses and that kind of thing. Mucus is good stuff, and especially good for protecting cells, actually, because it's a big, thick layer of viscous stuff that it's hard for like viruses and bacteria to penetrate through it. So it can protect your cells against viral and bacterial attacks. But cancer cells actually use it as camouflage. So when cancer cells start producing a bunch of mucin, particularly a mutated version that makes very short, that attaches to like these short little sugar chains, then your immune cells can't even really feel the surface of the cancer cells because they're buried underneath the mucus and they're just sticking sugars up which look normal to cancer cells. So this reactivating, heavy mucin production is a common way that cancer cells protect themselves from immune attack. It's hard when cells that normally don't produce mucus is making mucus. It's hard for white cells to even feel their surface. And even if they do, all they'll feel is just these normal sugars, which won't make them think there's anything going on there. Another relatively common way that cancer cells protect themselves is they stimulate the growth and activity of immunosuppressive white cells. You have what are called, one type of white cell you have is the regulator white cell, which are the white cells that normally shut down your immune system after it's done. So your immune system may detect like an infection with, say, bacteria or virus, and then it'll attack the bacteria and virus and destroy it. But then once the bacteria and virus have been cleared, you don't want the immune attack to keep going because immune attacks take up a lot of energy in the body. And, also if they get too vigorous, they can destroy healthy tissue. So you have these regulator white cells that normally shut off an immune attack after a few weeks. And they produce chemicals that shut down white cells and prevent them from moving around. But cancer cells can actually sometimes create chemicals that activate these regulator white cells and cause them to produce all these chemicals that shut down the immune response. And so that's actually a fairly common way that cancer cells evade the immune system, is they just kind of shut down all the local immune system by producing chemicals that activate the regulator white cells. Cancer cells are good at creating what's called microenvironments. That's these tumor environments that are very hostile to white cell function, very difficult for white cells to function in these areas. There's a lot of in tumors, there's a lot of very tightly linked cells. So it's hard for white cells to penetrate these balls of tightly linked cells and to feel around like they normally would. There usually isn't a lot of circulation around the inside of tumors. That tends to be an area without much blood flow. And white cells come from the bloodstream, so it's hard for them to get into the tumors. And inside the tumors, the cancer cells tend to produce a lot of these chemicals that are like a sticky web outside of the cell, um, which acts as a barrier to the penetration of white cells. And as in up here, these cells in tumors tend to produce immunosuppressive chemicals, just very similar to the chemicals produced by the regulator white cells up in number 4 here. Also, I didn't write it down, but tumors tend to have very acidic environments, and acid shuts down white cell function. So basically, cancer cells are good at stimulating anti-white cell responses and creating tumor environments that are really difficult for white cells to work in. Also, cancer cells tend to shut down what's called their MHC class I genes. These produce cell surface receptors called MHC class I proteins. And those proteins are necessary for some cells of the immune system, like cytotoxic T cells, to recognize and attack cancer cells. So if cancer cells aren't making MHC class I proteins, then cytotoxic T cells, for example, and also B cells won't recognize them, and so won't activate a response. Okay, so these are all different ways that cancer cells normally evade the immune system and protect themselves against attack. Some of the major ways are taking away their class I MHC proteins, which makes them pretty much invulnerable to regular cytotoxic T or B cell attacks. Those are different kinds of white cells that can attack cells. And most of them produce PD-L1 chemicals that shut down any white cell that touches them. And then there's usually some sort of either camouflage or barrier protection that cancer cells and tumors put up to prevent white cells from even getting to them. So some combination of these is used by most cancers to evade immune detection. And the cancers that become really dangerous to you are the ones that can evade your immune system, because the ones that can't generally get destroyed by your immune system. One problem with classical chemo drugs, those old style chemotherapy drugs, is they completely shut down your immune system. So when you're taking chemo drugs, you actually lose all of this protection that your white cells normally give you from cancers that haven't developed a great ability to evade the immune system. So you lose like an entire wing of cancer protection when you take a standard chemo drug. Another reason chemo drugs are not the best for dealing with cancer, that is standard chemo drugs. But the immunotherapy is actually designed to do the opposite of standard chemo drugs. Instead of shutting down your immune system, it's designed to stimulate your immune system and to get it to recognize and target the cancer cells. For most of the best targeted immunotherapies, what you need to do first, before you can develop the therapies, you need to identify which chemicals on the surface of your type of cancer are abnormal. And this is going to be different for different people and different types of cancer. So the surface chemicals that are abnormal in some types of cancer won't be abnormal in all types of cancer. But there are certain surface chemicals that do tend to be abnormal in a lot of different types of cancer. For example, growth hormone receptor genes, proteins tend to be abnormal, and those are surface proteins, either abnormal in number or in shape or both. So that's very common type of surface abnormality in cancer cells. And again, putting the PD-L1 chemical in their surface is also a common abnormality in cancer cells that evade the immune system. And taking away their MHC1 class 1 genes, proteins is another abnormality that's common. So there are certain surface chemical abnormalities that are, you don't just see in one type of cancer, you actually see it in many types of cancer. And every month we identify more of these abnormal surface chemicals in cancer cells, and that's good because those are the chemicals we can use to target immunotherapy. Okay, so what types of immunotherapy are there? Well, there's a bunch of different ones. There's antibody therapy. This was the first developed and is still the most common, where we take these proteins that are normally made by the immune system, antibodies, which are normally made by B cells, which are a type of white cell. And antibodies are designed to stick to one particular chemical and attract white cells to the cells that they stick to. And the white cells that they attract to that cell then will attack that cell in various ways to destroy it. So this is already a normal part of your immune system, but what we're doing here is we're redesigning some antibody proteins so that they will specifically stick to your cancer cells. One of the reasons your cancer cells survive as well as they do is that they actually don't allow the B cell part of the immune system to get active and produce antibodies against them. They cover up their abnormal receptors or abnormal surfaces, or they deactivate B cells using the PD-L1 system. So what we do is we find some chemicals that are common on the surface of your cancer cells, but not on the surface of your normal cells. And we deliberately create a lot of antibodies, what are called monoclonal antibodies, or mAbs, which just stick to that abnormal chemical on the surface. So these shouldn't stick to normal cells because normal cells have normal chemicals on their surface. These are designed to stick to abnormal chemicals. And when they do that, they can either just attract white cells, just stay there and attract white cells to attack those cells. But they can also do other things. For example, if the proteins that they're attached to on the surface that are abnormal are growth hormone receptors, these antibodies, if they stick to the abnormal growth hormone receptors, can actually prevent them from bonding growth hormone, which prevents them from activating cell growth and reproduction. The drug Herceptin, which was the first immunotherapy, is actually an antibody designed to stick to an abnormal growth hormone receptor on the surface of various cancer cells. It's actually on the surface of a number of different cancer cells, and prevent those growth receptors from activating and signaling cells to grow too fast. Another type of immunotherapy, which really hasn't gotten off the ground very much yet, is vaccines against cancer. The only one that's really, I think, been approved so far is the melanoma vaccine, which is a vaccine against melanoma skin cancer. It uses, what you do is you inject people with these abnormal chemicals that you tend to find on the surface of melanoma cells, which is a type of skin cancer cell. And then you do that so that the immune system will learn to recognize that abnormal protein and will mount a more vigorous attack against the melanoma cancer cells. Now, this doesn't always work because, again, these cancer cells have learned to protect themselves. But what this is supposed to do is generate a pretty vigorous immune response, which may be strong enough to overcome the natural resistance that these cancer cells have against immune attack. So this can work. It doesn't always, but it has been approved, for example, for melanoma treatment, where it works better for that type of cancer for various reasons than for a lot of other types of cancer. There's immunomodulators. Now, what these are hormones. These are cytokines and chemokines, which are white cell hormones, white cell communication chemicals that are designed to stimulate white cell reproduction, stimulate their movement, stimulate them to attack. Remember, some cancer cells protect themselves by essentially putting out chemicals that shut down the communication between white cells and shut down these stimulating chemicals. But if you add enough of these stimulating chemicals from the outside, you add extra, again, you can overcome some of that protection that cancer cells give by, preventing these things from getting made in the body. You can just inject them from outside the body, and so then they're there, and they stimulate white cells to grow and reproduce and move toward the cancerous tissue. So, that's helpful. By itself, it's, not going to be enough to get rid of the cancer, but that combined with some of these other treatments sometimes can put cancers in substantial remission. Two of my favorite therapies are what are called cellular therapies. There's a cell therapy where you take normal white cells from a person's body, like T cells and NK cells, and you re-engineer them to recognize and bond to and attack the cancer cells that you have. And then there's another one of my favorite treatments, viral therapy, where you actually re-engineer an existing virus. So you take a virus that's out there, you disable it in certain ways so that it's not going to, harm normal body tissue, and then you put chemicals on the surface of that virus that can stick to cancer cells. So just like these monoclonal antibodies, they're chemicals designed to stick just to these abnormal chemicals on cancer cells. And when the virus that's engineered with those receptors for cancer cell proteins, when they stick to the cancer cell, they get absorbed in the cancer cell, and then they start reproducing inside the cancer cell. That's what viruses do, is they're tiny little critters that get inside cells and reproduce inside the cell. They actually hijack the ribosomes of the cell to make more viruses. What's great about this kind of therapy is if you can get a virus just inside cancer cells but not normal cells, it'll reproduce inside the cancer cells and destroy them. And then once the cell's destroyed, the viruses break out of the cancer cell, thousands and thousands of them, and then go attach themselves to other cancer cells and get into them and destroy those cancer cells, and then reproduce inside them and break out into thousands more. And so this kind of response actually, unlike these other responses, gets stronger with time. These other things get weaker with time. That is, your most potent immunotherapy time for most immunotherapies is at the very beginning, the first few weeks after you start the therapy, and then it gets weaker and weaker and weaker. But the oncolytic virus therapy actually, because it's a real living virus that is just being reprogrammed to attack cancer cells, the response actually gets stronger and stronger with time as you get more and more and more viruses being created inside your cancer cells. So this has the potential to really be amazingly effective. The only thing that's limiting it right now, other than finding the right chemicals to put on the viruses so they attach to just the cancer cells, is that your immune system is designed to destroy viruses and cells infected with viruses. So what limits this therapy ultimately is your immune system attacking the engineered viruses. But we can engineer viruses so that they're more resistant to immune attack. course, when we do that, we make the cancer cells more resistant to immune attacks. So there's trade-offs here, is what I'm saying. But this one is exciting because at least it has the potential of being the only therapy that gets stronger with time, which means it could theoretically be the most effective of the therapies. And like here's a picture of an oncolytic virus. This is a virus that's engineered with receptors that bond to the surface of cancer cells. And so the virus gets into the cancer cells, reproduces inside them, and then you bust out even more viruses which attack more cancer cells and reproduce inside them, and the response keeps getting stronger and stronger, which is a good thing. Now, as far as the antibody responses go, let's see. So the antibody treatments you can do are either just plain antibodies that will stick to the surface of the cancer cells and attract like white cells to them. But you can also, when you engineer antibodies to stick to cancer cells, you can also attach poisons to the antibodies, like radioactive atoms that can destroy the cell with radiation, or toxic chemicals like chemochemicals that can destroy the cell DNA, for example. So we can engineer these monoclonal antibodies that only stick to abnormal chemicals on the surface of a cancer cell. Here's a cancer cell. But we can attach cytotoxic chemicals, which are cell poisons. These are the cell poisons we've attached to the antibody. So the antibody sticks to the surface of the cancer cell, then gets absorbed into the cancer cell, where these poisons are broken off from the rest. The antibody is broken down with proteases, and the toxins get released. And that, depending on the type of toxin it is, it generally kills the cell by damaging its DNA, and that causes the death of the cell. Theoretically, only the death of cancer cells, because this antibody should only stick to the abnormal chemicals on the surface of cancer cells. It shouldn't stick to normal cells, so it won't have this effect on normal cells. So that's a type of antibody treatment. And then we get to this last kind of treatment, what's called cellular therapy. And that's where they take white cells from your body, so your own white cells, but they re-engineer them to make it so that they will recognize and bond to chemicals, these abnormal chemicals that are only on the surface of cancer cells. And so, and the nice thing about these therapies is these chemicals that we put on the surface of the white cells when we re-engineer them, they can activate the white cell even without these other chemicals that would normally be required, like the MHC class I proteins. Most white cells can't be activated unless there's MHC class I proteins on the cells that they're bonding to. And that's true of like cytotoxic T cells. But when we re-engineer the cytotoxic T cells, we can re-engineer them with a modified receptor that doesn't require MHC class I proteins to activate. And so all that has to happen is the re-engineered T cell bonds to the cancer cell, and it will automatically activate and produce chemicals that destroy that cell. Probably easier if I show you this. So let's see which one of these, maybe this one. Okay, so normally if you have a T cell, in order for it to get active, the tumor cell would have to have an MHC protein on it. That's the only way that the normal T cell would activate. But a lot of tumor cells no longer make MHC proteins. They've shut off that gene. So they couldn't activate a normal T cell. But if you re-engineer these T cells with new receptors, what are called chimeric antigen receptors, or CARs, that's why they're called CAR T. They have these new receptors that don't require the MHC chemicals to activate. They just require the abnormal surface chemical, like the abnormal receptor that the tumor cell has, and they just need to bond to that. They don't need to all bond to MHC proteins. So these can kill cancer cells that protect themselves by taking away their MHC proteins. And we can get engineered white cells to bond directly to the tumors and then to be active even if the tumors don't have these all these proteins that would normally be required for activation. We can also re-engineer NK cells, what are called natural killer cells, which is a different kind of white cell, to bond straight to tumor cells without needing MHC or anything like that, to then kill those cells. There's advantages and disadvantages to each. For example, CAR T cells, they give you a really vigorous, powerful cell-killing response that takes a little while to ramp up, but eventually gets to a really vigorous state. Whereas the CAR NK cells, they actually start the attack sooner than the CAR T cells do, but after they bond to the tumor cells. But their attack isn't quite as vigorous or as powerful. But it is, and it's not as long lasting. The CAR T cells tend to maintain their attack for quite a while, whereas the CAR NK cells, they don't attack for as long. So what would be the advantage of CAR NK cells? Well, the advantage of CAR NK cells is natural killer cells. Is yes, it's true their attack isn't as vigorous and doesn't last as long, but they don't have one of the major side effects of CAR T. And one of the major side effects of CAR T engineered cells is what's called cytokine release syndrome, which is basically an overreaction of the immune system, where the immune system gets very overstimulated and starts attacking cells all over the body, not just cancer cells, but normal cells all over the body. And that reaction can actually become fatal relatively quickly. And that's a really bad reaction, and it's commonly seen with engineered CAR T cells, but you never see it with CAR NK cells. So what they're working on now is ways of doing CAR NK cell activation that allows the CAR NK cells to last longer, to live longer, to attack longer, so that their attack isn't time limited, and is more similar to the CAR T attack. But you still have the advantage that the CAR NK cells don't cause cytokine release syndrome. So you don't get that terrible side effect with CAR NK engineered cells. But anyway, these are very common now, and right now a bunch of these have been have been approved for the treatment of various kinds of blood cancers. So these engineered white cells are not that good at penetrating solid tumors. So they're not good for, normal cancers, which form big balls of cells, like, a breast cancer or a kidney cancer or a colon cancer. CAR T cells don't penetrate the tumors of those cancers very well. But in blood cancers where you don't have tumors, where the cells don't tend to aggregate, they're very good at killing the abnormal white cells. So when you have these blood cell cancers, we're talking about white blood cell cancers. So we engineer these white cells to kill other white cells, which they normally would never do. White cells normally do not kill white cells because white cells have MHC2 proteins, which prevent other white cells from attacking them. And with these engineered CAR T cells, they bypass the whole MHC system. And so even cells with MHC2 don't stop the attack of engineered CAR T cells. So these are very good because they don't have to penetrate through solid tumors, and they don't care about MHC proteins on the surface of the tumor. So these CAR T cells are great at treating things like leukemia, lymphoma, and myelomas. These are all blood cell cancers. And the CAR T cells are really good. They're so good that they're becoming sort of standard of care because they give people, much longer remissions and much longer lifespan who have those white cell cancers, which tend to be deadly very quickly. Like, leukemia, if you don't treat it, is usually deadly within a few months of symptoms. As soon as you start feeling bad and showing symptoms, these leukemias, these acute leukemias, are deadly within weeks to maybe 2 or 3 months. But when you treat them with CAR T cells as well as some standard chemotherapy, you can usually give people lifespans that, even if you don't do things like bone marrow transplants, you can give people lifespans in the, like 9 to 12 months, or even a couple to 3 years sometimes. So much longer remissions, much longer lifespans with the engineered CAR T cells when you're dealing with blood cancers. So far, that's really only true with blood cancers. But the other nice thing about these engineered white cell treatments is they can be used for autoimmune disease, because you make white cells that are good at attacking other white cells. You can target them not just to cancer cells, but to white cells that are attacking your normal tissue. That's how you get our autoimmune disease, is some of your white cells get misprogrammed and they start attacking normal cells in your body. And so you can engineer CAR T cells to attack those white cells that are attacking your normal cells, shut them down, and then they become really good treatments for autoimmune diseases like lupus or rheumatoid arthritis. So engineered T cells aren't just good for cancer, they're also good for other kinds of disease like autoimmune disease, which makes them really, really useful in medical treatment. Okay, so is there any downside to targeted immunotherapy? We've talked about the good things with targeted immunotherapy and how we can do it, and the fact that targeted immunotherapy has really significantly lengthened remission times and survival times for a number of different cancers, even just using chemicals that block the PD-1, PD-L1 association. These are what are called checkpoint inhibitors. We've been able to give people significantly longer remissions and lifespans with a number of different kinds of cancers. So targeted immunotherapies involve, as we've talked about, monoclonal antibodies that can stick to cancer cells and attract white cells to the cancer cells, or just deliver toxins or poisons to those, specifically to those cancer cells. We have white cell activator chemicals, which can, reinvigorate the white cell response against your cancers, although these you really have to combine with some other treatment here to get a good immune response. We can use nanoparticles that stick to tumor cells to deliver things like enzymes that can break down the proteins that are between cells. And that makes it easier for white cells to penetrate into like solid tumors. So this helps you with the treatment of solid tumors. We can use the checkpoint inhibitors, which stick to PD-L1 or PD-1, and prevent those two things from associating and deactivating the white cells. So this will cause reactivation of white cells that touch the cancer cells, and so will reinvigorate the immune attack against cancer cells. Many, many cancer cells produce the PD-L1. So this tends to work in a lot of different kinds of cancers, and it's really dramatically increased survival for many types of cancers. So some of these checkpoint inhibitors you've heard of, like Keytruda, Opdivo, you see commercials for them on TV. They're used now along with other chemotherapy drugs in many, many different types of cancer now to help people go into remission by getting white cells to attack the cancer. And we talked about viruses engineered to bond to abnormal chemicals on the surface of cancer cells, and that this has a theoretical ability to really be a great treatment because it's a treatment that gets stronger and stronger and stronger with time. So it's actually at the weakest when you first give the treatment, and then a few weeks or months later, it's actually an even stronger treatment as you get more and more of these white cells, of these viruses reproducing and killing more and more cancer cells. Again, what stops this ultimately is your immune system finally starts destroying these viruses. And sometimes that happens before the viruses can completely wipe out your tumors. So, that's one of the major reasons that this therapy, although it often gives good remissions, doesn't always give a cure, essentially a permanent remission. And then we talked about the CAR T and CAR NK cells and the advantages and disadvantages here. CAR T and CAR NK cells can bypass the MHC system. They don't need the cancer cells to have MHC proteins to activate, and that's good because most cancer cells don't make their MHC proteins. The CAR T attack is stronger and longer lasting, but the CAR NK cells are easier to engineer, and they don't risk some of the serious side effects of CAR T cells, like cytokine storms, or cytokine release syndrome, as they call it now. Okay, so what are the disadvantages of immunotherapy? Why doesn't immunotherapy always work perfectly? Always lead to essentially a permanent remission, which in the old days they would have called a cure. So let's talk about the disadvantages. Some of them are things you're familiar with from what we talked about before. But we'll talk about the one I already mentioned several times, which is some of these targeted immunotherapies. And it isn't just CAR T cells. Sometimes this happens with just antibody treatments, is you get an overreaction of the immune system, which leads to immune cells attacking a whole bunch of different healthy tissues, normal tissues, and destroying them. That's cytokine release syndrome. And that's a very serious side effect, which can be fatal. You also can get the other way. Some of these immunotherapies, like the viral oncolytic viruses, they fail for the opposite reason. Instead of this overreaction, which can be fatal, they cause the immune system to target the therapy, and the immune system actually targets the therapeutic cells and destroys them. So when you use viruses that kill cancer cells, you get an immune response where the immune system starts killing the viruses that's killing the cancer, and that ultimately makes that treatment ineffective. Again, we talked about how immunotherapies, because they're cell-based, can have trouble penetrating inside solid tumors. And that's because ultimately all immunotherapies are cell-based. Even the ones that don't use cells initially, like the antibody therapies, yeah, you can get antibodies inside solid tumors, but the antibodies are meant to attract white cells, and those cells just can't penetrate. And so that ultimately, most of the immunotherapies have trouble penetrating solid tumors. And most cancers have solid tumors, so that's a problem. Also, the chemicals that you use to target the immunotherapy, sometimes they're not as specific as you'd like them to be, and so you can get a chemical that you think is really unique to cancer cells, but it may not be. You may find a similar chemical like on the surface of different kinds of cells. And if that happens, then you'll get what's called OTOC effects, off-target effects, where the immunotherapy causes the immune system not just to attack the cancer cells, but also healthy cells in other parts of the body. And just like with regular chemotherapy, evolution is a problem. Cancer cells evolve by natural selection, and natural selection is triggered by anything that can harm or kill the cancer cell, anything that prevents it from reproducing. So, these targeted immunotherapies, they ultimately, if they're successful, ultimately they lead to the death of the cancer cell. And so cancer cells can evolve by natural selection. They can evolve resistance. Any cancer cell that has extra mutations that allow it to get around the immunotherapy, will live and reproduce and pass on those protections to the next generation of cancer cells. So just like with normal chemotherapy, evolved resistance of the cancer to the immunotherapy is a problem. One thing that we've discovered, though, is if you do two different kinds of immunotherapy at the same time, and where one kind is the cell-based, and the other kind is a chemical-based, like an antibody, for example, linked to a poison that sticks to the cancer cells, combined with a CAR-NK treatment, where the NK cells stick to the cancer cells. So this is a cell-based treatment. This is a chemically based treatment. It's using antibody proteins, but no cells. If you put those together, you usually get a pretty long-lasting remission, because it's a lot harder to evolve resistance to these two different kinds of immunoactivation therapies than it is to evolve resistance to just one of them. For example, if you use just the antibody therapy, cancer cells can evolve resistance to that pretty rapidly, usually within a few months, if that's all you're doing. But if you combine that with a cell-based therapy, then it's going to take cancer cells usually much longer to evolve resistance to those two therapies. And so you get much longer remissions, and sometimes even permanent remissions that way. Again, we don't use the word cure in cancer anymore. We say long-lasting remission, because you just never know if you've really been cured. Sometimes cancers reappear after 10 years of dormancy, so they don't like to use the word cure anymore. Another reason that immunological things tend to fail is when you activate the immune system to attack cancer cells. Your immune system is not designed to attack something forever. It's designed to attack something for a few weeks and then to stop the attack. You get the regulator T cells reproducing after a few weeks, and they start producing chemicals that shut down anti-cytokines and anti-chemokines that shut down the immune response, because it's dangerous in your body to have immune responses that last a very long time. And so the problem with these immunotherapies is you often get what's called immunological exhaustion, where the white cells just start shutting themselves down, or they get shut down by other white cells before they have finished destroying the tumors, the cancer cells. And so this, in addition to the evolution problem, you get this immunological exhaustion problem. And sometimes the opposite problem, the cytokine release problem, which is the overstimulation of the immune system. So you can get an immune response that's too vigorous, or one that's not vigorous enough, or that doesn't last long enough here. And finally, the good immunotherapy requires a lot of R&D, right? You have to figure out which chemicals are abnormal on the surface of your type of cancer. Then you have to find chemicals that can bond to those abnormal chemicals. And then you have to make antibodies that can bond, or you have to make, attach those bonding chemicals to cell surfaces, to make cells that will attach to the cancer cells. And so there's a lot of R&D that goes into that. But the good news is using AI-based drug design, AI is very good at figuring out and designing what chemicals that can bond to like the abnormal chemicals on cancer cells. And you can even use like global cellular RNA analysis to figure out which genes are active in a cancer cell and which proteins it's making right and which proteins it's not making right. So these techniques, which weren't around when I was in school, if they had been, I probably would have gone into cancer research instead of teaching. But these techniques have really shortened the time that it takes to find abnormal proteins on the surface of cancer cells and design chemicals that can bond to those abnormal proteins and to develop those chemicals. That kind of research used to take years and decades and long period of time, and it can now be done in weeks, sometimes a few months, but still an amazingly short period of time compared to how it was in my day. So this isn't as big a barrier as it used to be. Okay, but because of all of these things, immunotherapy is not perfect. It often leads to remissions. Remember, remissions are periods of time when the cancer stops spreading, when tumors stop growing, and when symptoms don't get worse, and sometimes get better because tumors may shrink. So that's remissions. Remissions don't always involve shrinkage of existing tumors, but they do always involve at least that the tumors aren't getting bigger. Often they also involve some shrinkage. And the symptoms of the cancer don't get any worse during that time. And sometimes, if the tumors shrink, they actually get better. So you feel less sick. So that's a remission. And nowadays, they measure everything in terms of remission potential and remission time. On average, how much extra time do you get in remission before the cancer advances and kills you? And, is the treatment effective for your kind of cancer at all? And what percentage of cancers can the treatment be used for, and that kind of thing. So, a really good immunotherapy can take a cancer that would normally kill you in a few months if you didn't treat it. And even with classical chemotherapy, will still probably kill you in 6 months or 8 months or whatever, and can extend your lifespan to a year or 2 years, most of which is symptom-free or with very reduced symptoms because of shrinkage of tumors and fewer working cancer cells. So immunotherapies tend to be pretty effective, and when they're combined with classical chemo or different kinds of immunotherapies are combined, they tend to be really effective. And we have seen cases where they've led to permanent remissions, which is what they used to call a cure. Where the tumors shrink to where you can't see them anymore, and in the period of time that they're looking, they don't see them come back. So they may be carrying out a study that goes on for 2 or 3 years, and in that time the tumors shrink, they disappear, they don't come back. Now that may be a cure, it may not, but like it may come back. There may be microtumors that may come back after the treatment is stopped. But still, that's a huge improvement in survival and quality of life over current treatments. So scientists, medical doctors, very excited about immunotherapies. I'm pretty excited about immunotherapies too. I think they have tremendous potential, not just for cancer, but also for like autoimmune diseases and chronic inflammation and other things. So, this is good stuff. So on that relatively positive note, I'm going to end this series on details of cell biology and modern cancer treatment, targeted therapies. Okay, so thanks for sticking around for the targeted immunotherapy, my favorite subject here in this overall kind of depressing series about cancer. Okay, so you take care.